This lecture is on blood cells. As I've told you before, with blood cells, then you have three types, and they are red blood cells, white blood cells, and platelets. And I'm going to go over each of these in turn. Then. So, your red blood cells, then, you recall this is sometimes abbreviated as RBC. These are your erythrocytes. And these account for slightly less than half of your total blood volume. Now, these cells are peculiar in that they have no nucleus, no mitochondria, and no ribosomes. You may recall from ANP1 that I said that a cell that has a nucleus removed will die. And that's what's going to happen to your red blood cells. Basically, your red blood cells are made, and then before they enter your blood circulation, the nucleus is basically taken out or spat out by the cell, and then the red blood cell enters your blood circulation. But once the nucleus leaves that cell, the cell is basically dying. So all your red blood cells are basically dying. They have a limited amount of time that they will be able to exist in your blood, and then eventually they will be taken out by the liver as having outlived their usefulness. So all of your red blood cells basically come with a use-by date, and what happens is that you keep making new ones to replace the old ones. So the red color in these cells then is due to hemoglobin molecules. Hemoglobin is what makes red blood cells red. It is also what allows your red blood cells to hold on to oxygen in order to transport it around your body. So, red blood cells, then, are flattened and thinner in the center than on the edges. So what did I I mean by that? What I mean is that if you were to look at a red blood cell from the side, you would see something, I'm exaggerating it, but you would see something that looks like this. It looks like a snow tube, basically, seen from the side. Normally, when you look at red blood cells in a microscope, you are seeing them from above. And in that case, what you wind up seeing usually is sort of a lighter center, and then a bit of a darker outside, kind of like this. So what you are seeing when you are seeing a red blood cell with what appears to be a light center and then the pink exterior like this, what you are seeing is the light from the base of the microscope coming up, here's your light, coming up from the bottom, but you only have a little bit of light going through the thicker part, whereas you have a lot of light going through the thinner part, and the result then is that you wind up seeing the red blood cell from above in a microscope with a light center and then the pink exterior. Okay, so what do these do then? Your red blood cells, you already know this. Their function is to transport oxygen and carbon dioxide, O2 and and specifically, they transport oxygen then from your lungs to your body tissues. And the red blood cells then also transport the carbon dioxide, which is made by your body tissues just as a part of being alive, to the lungs where you exhale it. Okay, so those are your red blood cells. Then we have white blood cells. You recall this may be abbreviated WBC, though their technical name is leukocytes. And these are all components of the immune system. And there are two major classes then. And this is based on appearance. This is strictly descriptive. Basically, what do they look like? So the two classes then are granular leukocytes, sometimes known as granulocytes. And this is just a descriptive name. These have what are called granular inclusions in the cytoplasm. Okay, what did I just say? You recall cytoplasm, that's the fluid inside a cell. So the point is, if you have a granular leukocyte, then inside the cell, in addition to the nucleus and whatever else it may have, you will see these dots, these things that look like grains. Those are the granular inclusions. Then the other kind of white blood cell major category then are your agranular leukocytes. You may recall A as meaning without. So these then are, by the way, they can be known simply as agranulocytes. And these are simply white blood cells that don't have granular inclusions. So these then are without your cytoplasmic granules. So yeah, in the early days of microbiology, a lot was done just by description. So it was, what color is the cell? Like red blood cells. Also, does it have these little grains in it? Okay, all these white blood cells that have grains in them are now known as granular leukocytes as opposed to agranular leukocytes. So as I said, these are very descriptive sorts of names. And likewise, even discussing the different types of granulocytes, then there are neutrophils. So these cells then are phagocytic. So they engulf items in your tissues and so these will be either pathogens or they will be debris. So the neutrophils will just go and grab things, basically. Now, you remember, I hope, phagocytosis. Again, phagocytosis is this general way that some cells have of attacking and eating other cells, and it's one of the ways that your immune system works. So if you wind up then having some pathogen, some invading microorganism or other, then you wind up having your neutrophils that will come along and basically engulf them and eat them and take care of them for you. So those then are your neutrophils. Then there are your eosinophils. Now, this is another case of a descriptive name. Eosin is a particular stain that's used in microbiology. And so these particular cells, phil means likes. So the eosinophils took up that eosin dye, and that's how they got their name. So your eosinophils are also phagocytic. And these engulf items in tissues that are labeled. With antibodies. So this is the other part of that antibody reaction, is the antibodies are released. They go out and they tag, basically, these invading microorganisms that they now understand. And then that, in turn, tells the eosinophils to go and eat those things. And another type of granulocyte, then, are your basophils. And these enter damaged tissues and release histamine and other chemicals. So this may have given you a little bit of an idea as to what those grains are about. So just as a quick aside, then, in thinking about your granulocytes, then, the granules, then, are tiny sacs. That contain enzymes. So, for instance, in the case of the phagocytic granulocytes, those enzymes will digest those things that have been engulfed. So, just looking briefly at the naming again, the neutrophils then have, as the name implies, neutral and subtle granules. The eosinophils have prominent granules that stain readily with the acid dye that is called eosin. So eosinophil, that is to say they will take up, they like that eosin. And then we have basophils. That have prominent granules that stain readily with basic, and by basic I mean alkaline, that is to say the other end of the pH scale from acid. These dye very readily then with alkaline-based dyes. So your granulocytes then have non-specific broad-based activity. Then we have the agranulocytes. And you have two different kinds of agranulocytes, one of which are called monocytes. And these enter tissues to become free macrophages, again, cells that will engulf other cells. So they will then engulf pathogens or debris. And I think I'm going to erase the other side of the board to give myself a little bit more room to write about the next variety then of your agranulocytes. So, still talking about agranulocytes now. We also have the type of white blood cell called lymphocytes. So these, although you find them in the blood, these are also cells of the lymphatic system. And these are the ones that provide defense against specific pathogens or toxins. So your lymphocytes are the ones then that do your specific immune system. you have lots of different kinds of lymphocytes. They have slightly different roles to play. They work together. But the upshot of the whole thing then is that these are your specific immune system cells, your lymphocytes. So they are the ones that will ultimately be making the antibodies and distributing the antibodies after they figure out what antibodies to make and everything else. And there are many steps in this process and many different types of lymphocytes that get involved. Now, one thing to keep in mind about this, so these are the cells that one way or another are responsible for your specific immune response. All the other cells that we've talked about that deal with your immune response are the ones dealing with general immunity. But here we are dealing with specific immunity, with specific antibodies that are released in response to specific pathogens or toxins. So one of the things to keep in mind, therefore, is that in the disease called AIDS, which is for acquired immune deficiency syndrome, it is basically a disease in which your own immune system ceases to work. What happens with AIDS is that there is a particular type of lymphocyte that HIV, the human immunodeficiency virus, or HIV attacks a particular kind of lymphocyte, not even all your lymphocytes, but a particular lymphocyte known as a T4 cell or a helper T cell. And the helper T lymphocytes are the ones that, kind of as the name implies, they help all your other lymphocytes to grow up and become functional. So without the helper T cells, it means that basically all of your specific immune response was shut down, which is why people, technically speaking, did not die of AIDS. What they died of was all of the diseases that moved in because they no longer had a fully functioning immune system. So it was not unusual, ultimately, for AIDS patients, for instance, to die of pneumonia. But as I said, that was simply an opportunistic infection, as were the cancers, as were the funguses, as were everything else that moved in. That is how AIDS patients wind up looking. All of that is simply because they no longer had an immune system that could reject all of those dangerous diseases. So anyway, those then are your lymphocytes. Those are your white blood cells. We're still talking about blood cells in general. So I am also going to talk about platelets. So these then are fragments. They're not even complete cells, really. So they're fragments of cytoplasm and enzymes. And these participate in the clotting response. And that's it for this lecture on blood cells.